Then it's all yours, Stephanie.
Okay, good morning everyone, and thank you for joining me for my thesis defense. Today I will be presenting my work entitled Relationship of Blood Parameters and Fertility-Related Traits with Age and Feed Efficiency in Young Beef Bulls. I would first like to thank my external examiner, Dr. Kim, for being here today, and Dr. Diane Patterson, the Graduate Studies Coordinator, as well as my advisory committee, Dr. Guillermo Cagnoli, Dr. Alma Ferdi, and Dr. Mariana Dialbiano. Throughout my presentation, I'm going to go through a typical scientific outline where I'll be describing both of my studies. So feed efficiency is an important trait for the beef industry as it decreases the environmental impact of the industry and also increasing its overall profit. But age is an important factor to consider, especially when evaluating young bulls that are going through sexual development, as age is associated with feed efficiency, body composition, blood parameters, and sexual development. And fertility-related measures have the potential to be used as indirect indicators of both feed efficiency and fertility. However, because these measures changes throughout the sexual development, care must be taken. And for example, when looking at blood hormones, you can see that testosterone tend to increase once bulls go through puberty. Looking at pixel intensity and scrotal circumference, both of these increases with sexual maturity, and scrotal circumference increases all the way up to adulthood. And then looking at semen quality increases in age, and you can see here that progressive motility drastically increased following puberty. And some of these fertility-related measures have already been shown to be associated with feed efficiency. These include scrotal thermography, scrotal circumference, testiculogenicity, semen quality, seminal proteomics, and testicular microscopy traits. So because all of these are associated with each other, there is possibility here to work on each of these parameters in order to improve the overall profitability of the beef industry. So if we were, for example, to have bulls that are both feed efficient and have good reproduction, we could potentially also produce progeny that also have good feed efficiency, which could lead to an increase in the competitiveness of the beef industry with other livestock species. And then having bulls that have reached puberty at younger age but also still have a good reproduction can lead to the production of daughters that also have good reproduction in earlier puberty, which increase the chances of reproductive success. And then if we're able to control and understand the effect of age on indirect indicators of feed efficiency, we may be able to use proxies for feed efficiency, such as blood parameters, which could greatly decrease the time needed and the labor needed to measure residual feed intake. So altogether, these could lead to a great decrease to the environmental impact and increase in profit of the industry. So it is known that animals that differ in feed efficiency also have differences in metabolism, and now there is more evidence also suggesting differences in reproductive function. And because both of these are related to each other, it is interesting to study them together. And age is also an important factor that affects feed efficiency, metabolism, and reproduction. Therefore, the hypothesis of this thesis was that animals that differ in feed efficiency and age would also differ in their metabolic and reproductive measures, as well as in their sexual development. The general objective of this thesis was to assess variation in blood parameters and fertility-related measures with feed efficiency and age over a repeated sampling period and on a specific date. So my first study, we investigated the association of blood parameters with age, feed efficiency, and sampling routines in young beef bulls. And this paper is currently under review for the Journal of Livestock Science. The objective of this study was to look at the variation in blood parameters such as blood hormones and blood metabolites with age and feed efficiency using two residual feed intake models, with one of them adjusting for age, over a repeated sampling period and on a single day measurement. For this study, we used 32 yearling crossbred bulls that were mainly Angus and Simmental of breed, and the bulls were housed at the Illawarra Beef Research Center. The bulls were fed a high moisture corn diet for 112 days, and daily feed intake was measured using automated feeding system. We evaluated body weight and body composition every 28 days, and the body composition traits that were assessed included rib area, back fat thickness, rump fat thickness, and marbling. Residual feed intake was used for the measure of feed efficiency. Our first model adjusted for body size and body composition traits, and then the second model that we used also included adjustment for age. We collected blood via jugular venipuncture at 8 different days during a 32-day sampling period. Then we also collected blood during a slaughter period where bulls were slaughtered in groups of 4 over 8 different days, and blood was collected during the bleeding process. Plasma was analyzed for blood parameters, including metabolic products such as carbon dioxide and acetate, ions such as potassium, proteins such as alkaline phosphatase and breeding soundness evaluation, and brain-derived neurotrophic factor, sexual hormones such as testosterone and luteinizing hormone, and metabolic hormones such as triiodothyronine, T3, and leptin. For the statistical analysis, we first assessed normality. Then we divided our bulls into age groups and feed efficiency groups using the 2 R5 models. And in both subcategories, we had 16 bulls. We did means comparison for the repeated measure using proc mix, and mean comparison on the day of slaughter, we used proc GLM. And in both cases, we adjusted for the breed effect. So now for some results. When we looked at testosterone, we observed lower testosterone in younger bulls, which agrees with the literature as testosterone increases as bulls go through puberty. The higher acetate observed in the higher CO2 observed in efficient bulls further supports the difference in stress response between feed efficiency groups, as blood acetates tend to decrease with an increase in respiratory rate after a stressful event. We observe higher acetate in feed-efficient bulls, and this may be due to the relationship between ruminal and blood acetate and feed intake, as higher level of blood acetate are associated with depressed feed intake in cattle. We also observe a higher testosterone in feed-efficient bulls. There's studies already that looked at that showed positive association between feed efficiency and testosterone. However, in this case, the difference may also be due to variation in age between the feed efficiency groups, as once we adjusted for age, there is no longer a difference. And then when we look at T3, we had lower T3 in feed efficient bulls for both R5 models. And T3 is associated with energy expenditure, and which may explain the higher feed intake in the inefficient bulls. And then looking on the day of slaughter, we still observe lower T3 in feed-efficient bulls, telling us that T3 is robust to variation in sampling routine. And then when we looked at brain-derived neurotrophic factor, this parameter was higher in feed-efficient animals for both models. And BDNF has been shown to be associated with feed intake and energy homeostasis, making it a potential indicator of feed efficiency for the future. In conclusion, blood parameters varied with age and feed efficiency, and once age was added to the model, some of the association that we observed differed. And the difference that we observed for breeding soundness evaluation in terms of feed efficiency were robust to variation in age, while the differences in T3 in terms of feed efficiency were robust both to variation in age and sampling routine. So during our first study, I observed interesting interaction between some parameters that are associated with sexual development. So my second study is investigating the relationship of age and feed efficiency with sexual development and fertility-related measures in young beef bulls. And this paper is currently under preparation to be submitted to the Journal of Animal Reproduction Science. The objective of this study was to look at the association between blood parameters and fertility-related traits with age and feed efficiency over repeated measures and on the day of the breeding soundness evaluation. For this study, we combined two populations of bulls, with the first one being housed at the Illawarra Beef Research Center and the second one at the Maritime Beef Test Station. And the bulls were yearlings and were classified as either British or continental. During this study, the bulls were evaluated at 3 different time points, and at each time point, we evaluated body composition, body weight, scrotal circumference, testiculogenicity, and blood hormones. And on the last day of the test, which was around the day of breeding soundness evaluation, we looked at CBC analysis, sperm tomography, and we collected semen for semen analysis. Bulls were fed for 112 days, and feed intake was measured using automated feeding system. The Maritimes bull were fed a diet that was mainly grass silage, while the Illawarra bulls were mainly fed a diet that was high moisture corn diet. For this study, our model adjusted for body size and body composition traits, as well as the different bull population. We measured scrotal circumference and pixel intensity of the testes because both of these traits are associated with testicular growth and development. And after we collected the images, we were able to analyze them by calculating proportionate square using the scrotal circumference of each bull, and then we measured the pixel intensity within each square. We obtained infrared images using an infrared camera, and then processed them in order to measure the total scrotal surface temperature and the scrotal surface temperature at the base and apex of the scrotum. And then at the base and temperature at the base and apex were used to measure the temperature gradient of the scrotum. Semen was collected using electroejaculation, and once the semen was collected, we right away we analyzed it for spermatility via visual assessment under the microscope. We also stained part of the ejaculate for sperm morphology and sperm viability, counting 100 sperm. And sperm morphology that were assessed include head defects, mid-piece defect, tail defects, and detached heads. And we also look at the viability. And we look at some of these sperm parameters in order to assess the breeding potential of a bull and also its stage of sexual development. And then for blood, we collected it through jugular venipuncture. And we look at hormones such as testosterone and also the complete blood cell count in order to have an idea of the health status of our bulls. For statistical analysis, we assessed normality, and then we divided our groups into age and feed efficiency, and bulls were divided into subgroups according to the distribution of age and residual feed intake. Means comparison was, for repeated measures, was done using ProcMix, while means comparison on the day of the breeding soundness evaluation was done using ProcGLM. In both cases, we adjusted for breed and population effect, and for the traits that are influenced by environmental temperature, we also adjusted for temperature humidity index. And then the traits, when we compared the feed efficiency groups, then we adjusted for age. Residuals were computed in order to calculate person correlation, and then the residuals were also used for partial least square analysis, where we computed 6 different factors that were used in partial regression analysis. So now for some results. So when we look at the performance, we observed lower body weight, lower feed intake, lower back fat thickness, and lower rib area in younger bulls. We also observe lower scrotal circumference in the younger bulls, which follows the literature, and then differently from other studies, we observe a trend toward higher pixel intensity in younger bulls. There was no difference in the blood hormones between age group, and differently to my first study, although the curves for testosterone appeared lower in younger bulls, there was no significant difference. And once again, on the day of breeding sinus evaluation, we still observe lower scrotal circumference in the younger bulls. In general, we observe higher scrotal temperature in the younger bulls, which may indicate a more active growing testicular tissue. And then in general, we observed lower semen quality in younger bulls, agreeing with the literature that semen quality increases with sexual development. When comparing feed efficiency groups, there was no difference in body composition traits, which is good, telling us that the model was properly adjusted. And we observed lower scrotal circumference in the efficient bulls, agreeing with one other study. And although there was no difference in the mean pixel intensity between efficiency group, we observed a trend towards lower pixel intensity in efficient bulls. And in general, this suggests that efficient bulls may have delayed testicular development. There was no difference in blood hormones between feed efficiency group, and differently from my first study, although the lines for T3 appeared different, there was no significant difference. And scrotal circumference was still lower in efficient bulls on the day of breeding sinus evaluation. We didn't observe a difference in pixel intensity on that day between feed efficiency group, but this is most likely due to the fact that most bulls at this time were post-pubertal. And in general, there was lower scrotal surface temperature in efficient bulls, suggesting lower testicular metabolism in these bulls, and that these bulls may allocate less energy toward testicular growth. When we looked at semen quality, we observed a trend towards significant for sperm head defects, and this agrees with other studies and may suggest differences in semen quality between feed efficiency groups. Looking at correlations between with T3, we saw positive correlation between T3, scrotal circumference, scrotal surface temperature, and sperm tail defects. And this further support the association of T3 with the testicular development and sexual development. When we look at the results for the variance analysis, looking at the whole population in terms of age, the most important factor explaining the variation in age was scrotal circumference. But then when we look only at the younger or older bulls, the main factor was semen quality. Then in terms of feed efficiency, the most important factor that was explaining the variation in residual feed intake was scrotum thermography. And then looking only at the efficient bulls, the most important factor was semen quality, while in the inefficient bulls, the most important factor was the complete blood count analysis traits. So in conclusion, the younger bulls presented characteristics of sexual immaturity, while efficient bulls displayed features of delayed sexual development. And scrotum circumference explained considerable variation in age, while scrotal thermography explained most of the variation in residual feed intake. And this suggests that scrotal thermography and scrotal circumference should be traits that are considered when testing young bulls in the context of feed efficiency. So in general, in both cases, for both groups, we observed differences in testicular development, and a lot of processes and organs work together in order for bulls to go through sexual development. These include the hypothalamus and the pituitary, which work together to increase the secretion of testosterone by their leading cells and increase the secretion of thyroid hormones by the thyroid gland. Bulls' secretion of testosterone tends to increase as bulls go through puberty. And the fact that in general, we observe lower testosterone in younger bulls may be linked to, is probably most likely linked to the differences in testicular development in these bulls. And we observe a positive correlation between T3, scrotal circumference, scrum temperature, and semen quality. And in general, we observe lower T3 in efficient bulls, which may be linked, and the lower T3 may be linked to the delayed testicular development observed in these bulls as well. And then for future studies, similar assessment that could be done over different physiological stages and over the circadian pattern of the bulls are needed. And also looking at the specific time of puberty and sexual maturity of bulls would be interesting, and looking at the microstructure and molecular aspect of other reproductive organs are needed. And altogether, this could allow the development of protocols that could help us identify bulls that are both feed efficient and have good reproductive potential. I would like to thank my supervisor, Dr. Yuri Montagnoli, as well as my committee members, the other academic support that I received, all my coworkers, and the technical staff at both farms, as well as all the funding agencies. And just to finish, over the past 2 years, I've worked extensively with beef bulls, but I also worked with producers in order to help them understand, help them with the purchase of their bulls. And I was lucky to be presenting, to do oral presentations at the Canadian Society of Animal Science in Ottawa at the 66 EAP conference in Warsaw, Poland. And I was really fortunate to get a scholarship with CMEX through ANSWER, and this allowed me to work in the industry. And then I'm really excited to be presenting my work in France this summer for the Association for Applied Animal Andrology. Thank you.